Hi, good morning, everyone. Thanks so much for the invitation to come and be back on a gym call. Yeah, it's great to see everybody and happy to come here. So I'm an assistant professor in the chemical engineering department of Delaware, although soon to be associate. My hopefully, fingers crossed, the board of trustees meeting next week announces that promotion. So I am recently assistant professor, and I'm here to talk about my recent NSF success. And I put it success in quotes because that's what I wanted to talk about today, the do's and don'ts of how I got to a success and experience with that. So I sort of feel like NSF success is somewhat of a misnomer, and that I'll hopefully share experience with that here today. So my research group is all interested in inhaled drug delivery, and there's a lot of challenges. We're maybe all familiar with this. We have, inhalers for asthma that are kind of ubiquitous, and everybody knows what they are, but they really haven't penetrated into other areas of respiratory diseases. I think COVID and COVID vaccines as a motivation for research. And some of the real reasons is you could make the best new medicine ever, but you really don't have a way to test it and know if it's going to work because the lung is super complex. And so being able to predict how things are going to work in the lung is a real challenge, which basically prevents new innovation in this space. And so when I started as an assistant professor in 2017, I was really drawn to this problem of thinking about new models of the lung. And we really are an experimental group. And so there's great innovations that happen in institutional or clinical sampling spaces. And we want to have a physical preclinical model that can enhance how we understand the clinical information, validate computational things, and have a high throughput way to actually get the information we want. So what is the information we want? Well, the first most important thing for an inhaled therapeutic is to know where it goes, right? Your lung is these huge, complicated, bifurcating structure. We want to know where it goes. And so that has these huge length scales that begin on the order of centimeters that go down to the microns. So once you get down into the actual structure of the lung, where you have cellular barriers and different mucosal lining fluids, that's when you can start to ask how that therapeutic is working and interacting with the cells. So it's a huge, complex problem from this micron to centimeter scale. And right now, the best way to go about this is to treat these two problems separately. If we could answer where therapeutics go, we can make much better predictions of how they're going to work because we would know what cells are there, how long they could stay there, and we could better predict what would happen inside the lung. All right, so when I started as assistant professor, this was my vision, was to create a full volume model of the lung that could actually test the entire dosage of an inhaled therapeutic. Take an inhaler, know where everything went. And so the information you can get from a patient is shown on this CT scan. So the opening at the top is the mouth, and then your lung starts to bifurcate. And from a CT scan, you can really only get about the first maybe 6 to 8 bifurcations. And everything else is just air and too small to be resolved because it's moving and the length scales are too fine. So how do you think about the rest of the lung when I said that we can only get to about 6 to 8 splits. Well, really, an adult lung is thought to have upwards of 23 or huge surface area of a tennis court that is captured in this physical space. And we want all of it because we want to know exactly where spatially these medicines are deposited. And so I'm an engineer. How can we think about, we can't make this, we can't recreate all of the structures of the lung. Even if we have a cadaver, we can't get the resolution of all those really fine features at the end. And there's millions of them. So how do we actually get the information we want, which is the spatial deposition profile, but in something we can physically actually make? And so our thought was to start with the important big features, the upper airway and the initial splits, and then discretize the lung into these 5 lobes. And then within each of these lobes, we could have sealed compartments that would allow us to have some relationship back to the spatial geometry. And then we could find something to go inside these lobes to approximate what the aerosol would have to experience. So it can impact on things, it can see surface area, it has to flow through these different flow profiles, it sees an expanding surface area, all of these things. But again, we can't make the tubes, we can't make those bifurcating structures. So we were inspired by mechanical engineers who can make lattices. These are essentially lightweight parts. They're porous media where they have a single unit cell that repeats uniformly in three-dimensional space, and they are really enabled by 3D printing. And so what I'm showing here, you can make all different sizes and shapes of these. Essentially, they're porous media. They're porous things, just like your lung, right? It's this open air, mostly open air space with defined features. And so we thought we can make these, and we're showing examples here, but we can go really to very fine resolutions that are closer to what it looks like in the lung. And so could we use these as an approximation for the aerosol transport that happens in the airspace? And so I'm going to cut to the chase and tell you my exciting story of, 5 years later, we actually are able to make this. So this is the very first prototype off the press, hot off the press last week. We have a 3D printed upper airway. We 3D print these lobe structures. We have these silicone caps on the end that exchange air with motors, and we can recreate breathing profiles. And then on the inside, we've populated them with these 3D printed lattice structures. So the top, we have only 2 regions in this model, but we can do a lot more in the future. The top is this open porous media that mimics the kind of larger features as they're continuing to split. These are larger ones, and then the peripheral becomes these really fine lattice meshes that you can't even really see through, but they're actually this porous media. All right, so I cut to the chase of this is the project, this is the vision, this is what I wanted to do, and I'm happy to say that we're making it. But I had a real trouble, and I still do, of where to get this funded, right? And so that's where, talking about my success today, yeah, you had the background of the project. Maybe you think like me, this is obvious, it's the lung, right? And so NHLBI, the Lung Institute, should really want this, right? And so I've submitted R01. They didn't like it. It's not a lung. It doesn't have the properties of a lung, and there's no cells in it. Cool. So I want it to be for inhaled medicines. I should be able to send that to the FDA, right? This is a regulatory thing. Pharmaceutical industry should want this. Oh no, they want it once it's validated, but it doesn't look like a lung enough yet. And we don't know if it's any work. We don't want to take a risk. Well, shoot. So what do I do? So NSF. This is more engineering. I'm an engineer. They should fund this. And so the cut to the chase. The first couple times I submitted this was rejected, and the success I'm going to talk about is how I did eventually get it funded. And I don't want you to feel bad for me, too bad, because I did end up hitting an R35 through the general medicine that has some elements of this idea in it. But it was really hard to be at this interface, multidisciplinary interface of something that is clearly has a medical focus. It's got pharmaceutical angles, but it's really fundamental, but it's not that fundamental. And so where do you find a home for this, and how do you actually get this supported? So the funding mechanism that I'm talking about today is the career proposal mechanism, which is an NSF early career award mechanism. And so this is the synopsis. It's supposed to be for early careers. You have to be untenured, and it's somebody who's going to serve as an academic role model in both research and education. And that's a key aspect of it, to have an integrated research and education. These are kind of key highlights for if anybody is interested or thinking about this award. It's 5 years, which is great, and having those big duration awards is really helpful for continuity. There's only one submission cycle a year. It hits in July, and you're only allowed 3 total submissions over the course of your pre-tenure time period. It's just you. They don't let any co-PIs on this, and you have to have, as I mentioned, this integrated research and educational plan. And so the research has to be this big career building thing that's they're going to support you for 5 years, but it should really launch a whole new area of research, which I can argue that this should. And then you have to have a lot of educational plans, both inside your institution and outreach, and how that integrates also with your research and research mission. So that piece requires a lot extra effort and extra thinking beyond just the usual level of research. All right, so if you're not familiar with NSF, as I was not, you have to figure out where you're going to submit your proposal first. So NSF first is divided at these directorate levels, and so I'm an engineer. I went to the engineering directorate. In the directorate, then there's divisions. And so then I am a chemical engineer, and so I went to the CBET division. Right, not there yet. Then there are different programs within CBET. And so, uh, unlike NIH, the review happens at the program level. And so the group that's going to fund it is also the group that's reviewing it. And so originally it's a lung, right? Submit it to the Engineered Biomedical Systems or EBMS division. And those were my two failures. So I submitted there twice, and I'll share more about that. Where I ended up getting it funded was in the particulate and multi-phase processes, or PMP group, which I didn't necessarily expect, and I think is part of the take-home message for today. All right, so my story on this. I did a lot of things wrong, and I hope in sharing you will learn from mistakes. So my first submission, I started as a faculty member in fall of 2017, and I think within 2 days of arriving, my colleagues are saying, well, when are you submitting your first career proposal? Because in engineering, it's expected that you're trying for this. And so I was finally worn down in 2018 and said, okay, well, I'll give it a try. My first time. I got 3 tries. I might as well give it a shot. So June 3rd, I attended the webinar for something that was due in later July. So already I'm starting on a bad foot of not starting soon enough. I went through my folders last night in preparing this talk. I started a brainstorming document July 4th. And so pretty much my memory of this summer is all of July sitting and working on the proposal, having really hurried, rushed meetings, trying to get some pieces together, and submitted a proposal. It was one of my earliest proposals I submitted, and, I didn't know what to expect. So July submitted, I, start hearing and seeing other people getting career proposals awarded in December, January, February, and I haven't heard anything. And so I'm like, well, maybe that's a good thing. I'm talking to my senior colleagues, and it's not until April 8th that I get the official decline. So maybe I was on the cusp, maybe something was good. A few months later, I get the reviews. And so in NSF, you get 4 individuals who review your proposal, and they give you a score. It's either excellent, very good, good, fair, or poor. So 5 categories of descriptors. And then the panel discusses that together, and you get a either non-competitive, competitive, or highly competitive delineation. And so these were my reviews. The first time I got 2 very goods straight out, and then I got 2 of these very good/goods. And so it was ranked competitive, and I was overall really jazzed, right? My first time through, I did all the bad things that you're not supposed to do when submitting a proposal, and I got some comments that I thought I could address, and the comments were very addressable. I didn't describe enough of the kind of fundamentals behind the lung, and they were things that I thought I could really address. So I did much better the second time, and I turned it right around. So I found out in April. By May, I was already going through the feedback. I started regular meetings with mentors, going over drafts. I found documents from the middle of June where I had a really fleshed out outline. I'd restarted things. I had checklists of how I was going to enhance everything, and I resubmitted in July. And so based on the prior year, again to the same EBMS group. And so based on, prior things, I wasn't expecting to hear anything until April, and maybe something, I'd hear something positive sooner. Early December, I just got a straight out decline. And that was really like, what? Come on. And I still had a very good, but I went down. So I had more of these very good/goods, and then 1 good outright who, so after this, the panel still said it was competitive, but my scores are going down. And if I look at these 2 proposals, this proposal number 2 is much better. I did lots of things wrong in the first proposal, and this one is arguably much better. So I was really sad and kind of pissed off too, right? Because I had submitted a better proposal and I got worse reviews. And so it was really, I had to step away from it. I decided I wasn't going to submit right then in 2018. I was going to wait a year. And in waiting a year, then it was COVID, and I felt like I was still in the same place. And so I really didn't think about resubmitting for a while. And when I would go back to these reviews, what it kept coming back is this is not a lung, there's not cells in it, kind of the same sorts of reviews I got from H. I started submitting other things, and some of those rejected FDA and NIH proposals came in this time too, and so I'm really like bummed on this project. What we're doing at the time in the lab is still working on it because I had some other funds I could support people and students on a fellowship. And what we were spending a lot of time on was the fundamental aspects of this work. So no one has ever studied aerosol transport in these lattice materials. And so how do you pick the right one was a question that we were stuck on because there was no underlying theory to say, if you pick this lattice, this will happen. And so we had to go back to our chemical engineering roots and really study both airflow and particle deposition in these, and started publishing papers in this area. And it became an interesting problem because we never really, there was no theory. We had these new materials that no. So I finally decided last year in 2022 that it was my last chance. I had to submit it. I wasn't going to get it, but I needed to do it just to know. And it was going to be okay when I didn't get it, accepted, because I had some other funding that hit, and the project would live on. And so I just was going to do it to see if it happened. And so what did I really want to do? Well, when I thought about what I really wanted and what we needed more understanding of these fundamentals happening within the lab. And so I also waited till kind of June to decide, I'm going to do it. And I started now thinking a lot more about the background, fundamental engineering sides of things. Submitted the proposal in July. Early November, I didn't think I'd hear anything. I got an email from the program director who asked me to rebudget and said, we're considering funding your proposal, but you asked for too much money. Can you find a way to get it to this target number? I did this very quickly and wrote an abstract at the beginning of January. So these are my scores, and I was given a highly competitive review from. And so mostly to take in mind, it's a different panel, which is also a rule that when you're submitting this, everyone says pick a panel and go to that same panel always. But when I was looking at the reviews from the prior panel and who got funded from those, it was people who were making tissue engineered models that had cells and tissue level complexity. And so when I would get these reviews of your thing doesn't have cells, or how are you going to consider the cellular aspects of it? Well, that's something my lab does. It was not what I was focusing on in the proposal because there were so many other things to do instead. And so this proposal that actually got funded is all about fundamental aerosol transport through these weird lattice materials. And the very last sub-aim of the proposal is, and then we're going to put it back in our lung model that we're working on. And so it's much more about fundamental science, advancing cool new materials that have broad applications, and that's how I sold it. But the application that we cared about was the lung, and I did go pretty hard on the proposal there too. And if you read so it's funded, it got a nice feature about this, and it's to study 3D printed lattices for testing inhaled medicine. And it's really to study aerosol transport in 3D printed lattices, period. And then there's just a tiny little bit of, this is our application. So we really had to think thoughtfully about how to tailor this to the organization to get the aspects that we needed, covered. And so my take-home message to you is, think about how you can find something really fundamental that the program will care about. Find a program director who you can talk to and really, pick their brain whether that's going to fly at their panel. Try to review if you can, because that makes NSF is really wanting reviewers, and it's much easier to get on review panels. And that's how I realized that I was on the wrong, the EBMS was the wrong place for me, but the particle multiphase would, be really excited about this. I guess the last piece is always be persistent, because usually when, in my experience, you write a proposal that you don't think is going to get funded, and you just write what you want to do, those are the ones that end up getting funded, and the ones that you spend a lot of time on and you think are perfect and beautiful, and of course they're going to fund them, then they never happen. So I say hang in there and be persistent, and eventually something will hit, and all of the energy of writing it, isn't wasted because you repurpose. Hang in there. if anyone's interested in any of these things, my group does a lot of immune engineering stuff. We do cellular things. We also have this model that we're excited about. And I'd be happy to talk about research or happy to talk about career proposal in general and share my proposal. So if you're thinking about writing it in the future and you're interested in seeing mine, I'm happy to share that with anybody because there's a lot of other documents. So thank you. I'm going to stop sharing so I can see the screen again. And yeah, happy to answer any questions or chat about it.